Hello students, a warm welcome to all of you in ITLS Academy. In this video lecture, we will discuss the DNA fingerprinting. Before understanding the DNA fingerprinting, first we discuss little bit about the DNA. What is the DNA? DNA is a deoxyribonucleic acid. Okay, it is a genetic material. It is a genetic material which easily transfer to parents to their offspring. This is a biomolecule which easily transfer to parents to their offspring. That's why this biological molecule is known as a genetic material. Okay, if we're talking about the composition of DNA, it contains a three component. First is a sugar molecule, second is a phosphate group, and third is a nitrogenous base. ATGC, adenine, thymine, guanine, and cytosine. These are the four nitrogenous bases. Okay. If we're talking about the composition, what now we're talking about the genome. What is a genome? Genome is a completely genetic material of any individual. Genetic material of any individual. If we're talking about the human genome, that in human genome, if we're talking about the human genome, then we talking about the complete genetic material of humans. Okay, in 99.99% of genetic material in all individuals are same. This 99.99% of genetic material or gene or genome our in all the humans are same, but we but, but we all are different from each other. Why? Why we are different from each other? We are different from each other due to the 0.1% of our genetic material. Only this 0.1% genetic material of one individual to another individuals are different. That's why we all are differ from each other. In DNA technology, in DNA fingerprinting, we also be study this only this 0.1% part of our DNA. In in DNA fingerprinting basically in DNA fingerprinting process we will study the STR, that are the short tandem repeats. In STR, there are two types of STR. There is a types of this is a types of a repeated unit, repeated unit of this nitrogenous base. Okay, STR, short tandem repeats, and also known as the satellites. Okay, satellites. What is a satellite? Satellites is also the repeated unit of nitrogenous base. These satellites are two types. First is a microsatellite and second is a mini satellite. Mini satellite in DNA fingerprinting, we study the mini satellite. What is a mini satellite? Mini satellite is a AT rich region. It contains 10 to 100 repeated unit of it 100 base pair repeated unit of base pairs. It repeats 50 500 to 3300 times. Okay? In DNA fingerprinting, our target to study the mini satellites. Mini satellites are the AT rich regions. Okay? Now we understand the DNA. Now we understand the DNA fingerprinting. What is a DNA fingerprinting? DNA fingerprinting is a chemical method. What is a DNA fingerprinting? Is a chemical scientific chemical method which used to determine an individual's genetic makeup or if relevant to compare it to the other. In DNA fingerprinting, this technique is applied to the human genome's polymeric regions. What is the polymeric regions? Polymeric region, nothing else, it is a mini satellites. Due to this polymeric region, we all are different from each other. DNA fingerprinting is a chemical method in which we determine the genetic makeup of any organisms and it compared to the other one. Okay. This DNA fingerprinting is also known as a DNA profiling, molecular typing and molecular fingerprinting. Okay. More than 99 per human genome is identical across the all individual. Only this polymorphic region is varies. Okay. Now we understand the history of DNA fingerprinting. In 1984, Sir Alex Jeffrey discovered this unique technology in the laboratory of University of Leicester at the genetic department. After the 1844, in 1988, the scientists of CSIR CCMB Hyderabad used and developed the different probe. That's why the India became a third country in the world to develop its own DNA fingerprinting probe. After the 1988, in 1991, the Kerala High Court accepted the DNA fingerprinting report as a evidence to resolve a paternity case. Paternity dispute. Now we understand the principle of DNA fingerprinting. This DNA fingerprinting refers to technique to obtain a multiple band of DNA by different size by the digestion of DNA with restriction and enzymes and hybrid it with specific core probe and compare it with the DNA mapping to another individual to evaluate the similarity. The principle of DNA fingerprinting says that first we isolate the DNA, then we fragment out the DNA, then separate the DNA, then hybridization, then autoradiography, and finally we obtain our result. Okay, now these are the basic steps of DNA fingerprinting. First is a sample collection, second is the DNA extraction, third is a PCR and restriction, the treatment of restriction endonuclease, then gel electrophoresis, then Southern plotting, and finally autoradiography. These are the basic step of DNA fingerprinting. Now, first we understand the procedure. Procedure में first step है हमारा sample collection. First we collect the sample, and these samples are blood, hair, saliva, semen, body tissues, DNA sample have are obtained from the vaginal cell transfer to the outside of the condom layer during the sexual intercourse. These are the some samples in which by the help we these are the some samples by the help of this sample we easily isolate our DNA and perform further process. After after the sample collection, we isolate the DNA. There are a lots of methods. Basically, first is a manual method and second is a kit method. Manual method is a long method and kit method is a very easily and very quick method. By the help of this two method, we easily isolate our DNA. After the isolation of DNA, we will perform the PCR. PCR is a polymorphic chain reaction. In PCR, there are three main steps. First step is denaturation, second is annealing, third is extension. In denaturation step, our double stranded DNA convert into the single stranded DNA. Okay, here. And the required temperature to perform this step is 95 degrees centigrade. After the performing the denaturation process, this is the double stranded DNA. Now our DNA will single stranded. Okay, and the hydrogen bond between these bands will break. Okay, now we perform the annealing process. For the performing this annealing process, the required temperature is 55 degrees centigrade. In this process, the forward and reverse primers are binding with the DNA templates. This is a primer. To perform this process, we use the two types of primer. First is a forward primer, second is a reverse primer. Forward primer will help the synthesis of DNA at a forward in a forward direction. In reverse direction, the reverse primer help the synthesis of DNA in reverse direction. After the annealing process, the third process is extension. In this step, our DNA will amplify, okay, or synthesized. Now, after performing this PCR process, we found the two bits of DNA. If we have a single band, after the first cycle, this converted into a 2 DNA. Okay. After the second, it will convert into a 4 DNA. After 8 DNA, then this formula will apply. Okay. N is the number of cycle. If we perform the 5, the number of DNA molecules is 2. After the performing the 5 cycle, we found the 1 DNA sample. We found the 32 DNA sample with only one DNA. If we have only one band of DNA, then after performing the 5 cycle, we obtain a same copy of 5 DNA. That's why this PCR can use to amplify the DNA. Okay, these are the PCR component. First is the isolated DNA sample, then primer, nitrogenous base, या फिर nucleotides, Taq polymerase here used as a DNA polymerase. This is used for the synthesis of DNA. Now nitrogenous mixer and PCR tubes. We put all these components in this PCR tube, and after this we put this PCR tube in a thermal cycler. In this thermal cycler, these three steps was performed. This is all about the PCR. Now we understand the treatment of DNA sample with the restriction. What is the third? Treatment of DNA sample with restriction endonuclease. What is the restriction endonuclease? Restriction endonuclease is a types of enzymes in which is cut the DNA at a specific site. Okay, it cut the DNA. It cut the DNA at a specific site. Okay, now the isolated DNA, okay, is mixed with the restriction enzyme to be create the fingerprints. Okay, because this restriction endonuclease cut the specific site of DNA and convert the whole genome into a small segments. Okay, that's why we use the restriction endonuclease. Okay, the restriction enzyme will cut the DNA in a pattern that will form a different from DNA to the other sources unless identify the DNA is the same. Matching known or unknown sample enables to identification. Okay, now DNA finger produced by the restriction enzymes are separately by the size using the gel electrophoresis. After performing this process, after treating the DNA with endonuclease, we separate out these bands with the help of gel electrophoresis. Now we understand the process of gel electrophoresis. Gel electrophoresis is a technique commonly used to separate the biological molecule based on a chemical characteristics such as a charge and polarity. What is a gel electrophoresis? Gel electrophoresis is a separation technique of biological molecules. Such as RNA, DNA, and proteins, and these molecules are separated on the basis of their charge, size, and polarity. But for the separation of DNA, we will use the agarose gel electrophoresis. Okay. For the isolation or for the separation of DNA, we use the agarose gel electrophoresis. Okay, now we understand the procedure of basic steps of gel electrophoresis. First, we prepare the agarose buffer solution. For the preparation of this agarose buffer solution, we require the agarose. Okay, agarose is a solidifying agent. Okay, after the preparation of agarose and buffer solution, we poured this agarose buffer solution into a plastic tray. Okay, and also placed a comb at the one end of the tray like this. And after the cool down of our solution and after the solidify the agarose gel and this gel, after the solidification of this gel, we remove this comb. After the removing this comb, we easily see these wells. This is known as a sample wells. In this sample wells, we put our DNA. Okay. Here, we add the tracking dial with our DNA sample to track the DNA. Okay. After the whole process, we connect it with the electricity. Here is a negative charge and here is the positive charge. Here is the cathode, here is the anode, and our DNA in our DNA is a negatively charged molecule. That's why it can easily move to positively charged anode. Okay. After this, after the electricity after this, these bands are easily separated on the basis of their size. Small bonds can easily move fast and large bands move slow. That's why here this separation is based on their size. Okay. After performing this process, we found these types of band. Okay. After the performing this process, now we transfer this band onto the nylon sheet because this sheet is very it is basically a jelly like structure. It can break easily. That's why we transfer these bands onto the nylon sheet or the nitrocellulose membrane. After performing this process, we perform the Southern plotting. So now here we transfer the DNA bands onto the nylon sheet. Okay, this is a whole equipment. Okay, here is a port sheet, rubber band, Wattman paper. Then this is the Wattman paper. Then we put our nylon sheet. Then we put our band. Okay, then we put here some weight. Here the transfer process is based on a principle of capillary action. The transfer of DNA sample from gel to the nylon sheet is due to the capillary action. Okay, and due to this weight, this is a buffer. Due to this buffer, this capillary action will perform easily and our DNA is negative charge and our nylon sheet is positively charged. That's why this DNA will easily transfer on this nitrocellulose membrane or nylon membrane. After performing this whole chapter, we incubate it for a 24 hours or overnight. After the overnight, we use some radio labeled probe. These radio labeled probes are the complementary sequence of our desired band. Okay, we pour the heavy after the transferring of DNA into the nylon sheet, we perform the probing or hybridization process. Okay, इसमें we adding the radioactive and colored probe to the nylon sheet, which is a complementary to our the target sequence. Okay, each probes were only stick on a one or two specific placed on a sheet. Okay, after providing the probe, hybridization process will perform. Okay, this जो हमारे radio level probe can easily hybridize with their complementary sequences onto the nylon sheet. After the performing this whole procedure we wash out the membrane to remove the non-specific binding and clear the background. After the whole this process, we perform the autoradiography. On the autoradiography, we use the X-ray film. We hybridize the membrane with the X-ray film and easily diagnosed our result, and we obtain the band like this. Okay. For example, B1 shows the victim sample. Okay. S1 is the sample of criminal site. S1 is a suspect, second S2 is the second suspect, and third is a sample of third suspect. Okay. Now here we perform the matching. First to first, second to second, third to third. After the matching the process, we predict our result easily. We analyze that the sample of criminal site is matched to the which one of this subset will suspect. Okay. Now, for example, any fingerprinting. Okay. Now we understand the application and role of fingerprinting in different sectors. This for the assessment of inbreeding, pedigree analysis, immigration process, plant variety identification, bioconservation, wildlife life science, establish the paternity and maternity dispute and identification of missing child, and some fields and some areas in which the DNA fingerprinting have a lots of role and lots of application. This is all about today's lecture. If you like my lecture, then follow us on Instagram, Twitter, YouTube, WhatsApp, LinkedIn, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share, and subscribe ITLS Academy. Thank you.